Monoclonal antibodies are also known as mAbs. They are large quantities of identical antibodies produced by a clone of genetically identical plasma cell. The concept of monoclonal antibodies came from the idea of magic bullets, first proposed by Paul Ehrlich in 1906. Magic bullet is a concept wherein he thought that there will be something that will produce substances that could seek out specific disease-causing agents. He won a Nobel Prize in Physiology or Medicine with Dr. Eli Mekinoff. Production involving human-mouse hybrid cells was first described by Gerald Schwaber in 1973. However, Kohler and Milstein was the one successful in making fusions of myeloma B cells to create hybridomas that could produce antibodies specific to known antigens and that were immortalized. They won Nobel Prize in Physiology and Medicine in 1984. With monoclonal antibodies, we have to remember that antibodies have a high degree of specificity. Therefore, if they are specific, they could be useful for diagnosis and treatment. But long time ago, we didn't know how because of two things. One, B cells that divide by mitosis do not produce by antibodies. On another hand, plasma cells that secrete antibodies do not divide. And in order for this problem to be solved, what they thought of in 1970s is that they should put plasma cell and cancer cells together. And by cancer cells, they specified myeloma, which is a cancer of plasma cell. By combining the two, they are able to create a cell known as hybridoma. The hybridoma will gain the ability of both plasma cell, which is to produce specific antibody, and cancer cells' ability, which is to divide endlessly. In order to do this, there should be a defined standard operating procedure in producing monoclonal antibodies. All the images that you would be seeing are for diagrammatical purposes only. This is not how it looks like in real life, 100%. So for the production, first and foremost, a mouse or other organism is injected with an antigen that will stimulate the production of the desired antibody. B cell in the mouse that recognize the antigen divide to form a clone of plasma cell able to secrete an antibody against it. A cancer cell is taken from a tissue culture. B lymphocytes that were taken from the mouse spleen are fused with cancer cells to produce hybridoma cells. To identify the hybridoma cells that produce the desired antibody, individual cells are separated out. Each hybridoma cell is tested to see if it produces antibody on exposure to the antigen. Any that do are cultured so that they produce a clone of cells all secreting the desired antibody. As we've said earlier, monoclonal antibodies are useful for two things. First one is diagnosis and two is treatment. Diagnosis can include locating position of blood clots in a person with deep vein thrombosis, wherein instead of a random antibody, the mouse is injected fibrin, a radioactive chemical that produces gamma radiation is attached to each antibody. Once introduced into patient's blood, the monoclonal antibodies are carried around the body in the bloodstream and binds to any fibrin molecules with which they come to contact with. Radioactivity emitted is detected by gamma ray cameras, and this helps in locating the position of the labeled monoclonal antibodies. Another helpful use of mAbs in diagnosis is locating cancer cells, identifying exact strain of virus or bacterium, blood typing before transfusions, and tissue typing before transplants. Once these monoclonal antibodies are used for treatment, it is required for them to be humanized. Antibodies from non-human species have been modified to increase similarity to variants found in human, and the one being modified in these antibodies is the protein sequence in both the heavy and light chain. There are two ways to humanize antibodies or monoclonal antibodies. That is to 1. Changing type and position of sugar groups attached to heavy chains to same arrangement found in humans. And 2. Alter the genes for heavy and light polypeptide chains into human sequence. There are 4 different examples that were stated in the book. First example of a humanized monoclonal antibody is
trastuzumab,
which is one of the breast cancer treatment, and this one comes from the mouse. This one binds to receptor proteins produced in abnormal quantities in the cell surface membrane. This antibody will mark the cell for destruction. Another example is called as
ipilimumab,
which is a cancer therapy for melanoma. This binds to protein produced by T cells that reduces immune response.
infliximab,
This one is used for rheumatoid arthritis and binds to a protein secreted by T cells that causes damage to the cartilage in joints and blocks its action. This one is required to be done in a monthly or bi-monthly intervals. And last but not the least, we have
rituximab,
rituximab, which is used to control B lymphocyte that leads to cell death. This is used in leukemia, multiple sclerosis, and rheumatoid arthritis. Trials on myasthenia gravis with the use of this monoclonal antibody has been successful.